Unit 10 Animal Cell Culture and Growth Kinetics, which is an important topic in biotechnology GAT-B 2026 aspects. It mainly deals with how animal cells are grown outside the body in laboratory conditions and how their growth is studied scientifically. This unit is very important for GAT-B because many conceptual and application-based questions come from here. In this unit, you will learn both practical and theoretical aspects such as media preparation, cell growth behavior, and advanced topics like cloning and transgenic animals. Overall, this unit connects basic biology with industrial and medical applications. So, understand it clearly will help in both exams and future research. This slide explains how BioBrainz platform supports students preparing for GAT-B 2026. It focuses on concept-based learning, which means understanding concepts and topics clearly instead of just memorizing. It also mentions that practice questions like MCQs and previous year questions are provided, which are very useful for exam preparation. Additionally, it highlights features like mock test, doubt solving, and regular updates. These tools help students track their preparation and improve weak areas. Overall, the slide is more about preparation strategy rather than scientific content. Animal cell culture means growing animal cells in laboratory conditions outside the body, which is called in vitro. These cells need controlled conditions like proper nutrition, temperature, and pH to survive and growth. There are different types such as primary culture, secondary culture, and continuous cell lines. This technique is widely used in vaccine production, drug testing, and genetic engineering. However, there are some limitations like contamination risk and genetic changes over time. So, maintaining sterile conditions and proper monitoring is very important in cell culture work. Culture media is the liquid or gel that provides nutrition for cell growth. It contains inorganic salts for maintaining osmotic balance, amino acids for protein synthesis, vitamins for metabolism, and glucose as an energy source. Common media include MEM, DMEM, and RPMA 1640. Serum, especially fetal bovine serum (FBS), is often added because it provides growth factors and hormones. However, serum has drawbacks like variability and contamination risk. That is why serum-free media is preferred in industries, as it gives more consistent and reproducible results. Animal cells require specific conditions to grow properly. The temperature is used mainly at 37 degrees Celsius for mammalian cells. The pH is kept around 7.2 to maximum 7.4 using a CO2 incubator, which helps in maintaining the buffering system. Other factors include oxygen supply, osmolarity, and sterility. Proper sterile conditions are maintained using laminar airflow and antibiotics. When cells grow too much, they are subcultured or passaged using trypsin, which helps in maintaining healthy cell growth. Cells can be stored for short or long periods depending on the requirement. For short-term storage, they are kept at 4 degrees Celsius. For long-term preservation, cryopreservation is used, where cells are stored in liquid nitrogen at minus 196 degrees Celsius. Cryoprotectants like DMSO and glycerol are added to prevent ice crystal damage. Cells are cooled slowly and thawed quickly to maintain viability. After thawing, tests like trypan blue staining are used to check if the cells are live or dead. Anchorage-dependent cells need a surface to attach for growth, such as fibroblasts, and they grow as a monolayer. In contrast, anchorage-independent cells like lymphocytes grow freely in suspension without needing any surface. These differences are important for their applications. Adherent cells are useful in tissue studies, while suspension cells are used in large-scale production, like antibody manufacturing. These classes is very important for exam questions. Cell growth occurs in different phases: lag phase, adaptation; log phase, rapid growth; stationary phase, nutrient limitation; and decline phase, cell death. Among these, the log phase is most important because cells divide rapidly here. Two important concepts are doubling time, time taken to double the population, and growth rate. These depend on factors like nutrient availability and waste accumulation. Understanding the concept is very important for graph-based questions in exams. Microcarriers are small beads that allow adherent cells to grow in suspension. This helps in increasing the surface area, which leads to higher cell density. Materials like dextran and collagen are commonly used to make these beads. This method is useful for large-scale production such as vaccines and recombinant proteins. However, problems like shear stress and bead aggregation can affect cell growth, so proper control is required. Macrocarriers are large structures like fibers and sponsors that support cell growth. They help in mimicking a three-dimensional environment, which is closer to natural tissue conditions. These systems improve cell differentiation and productivity, making them useful in advanced research. However, they have limitations such as difficulty in nutrition diffusion and complex scaling up. Hybridoma technology is used to produce monoclonal antibodies, which are identical and specific antibodies. It is done by fusing B lymphocytes, which produce antibodies, with myeloma cells, which can divide continuously. This fusion creates hybrid cells called hybridomas that have both properties. The process includes immunization, cell fusion using PEG, selecting using HAT medium, and screening using ELISA. These hybridomas are very useful in diagnostics and treatment, especially in diseases like cancer. This topic is very important for GAT-B exams. Stem cells are special cells that can divide and differentiate into different types of cells. There are three main types: embryonic stem cells, pluripotent; adult stem cells, multipotent; and induced pluripotent stem cell, iPSCs. These cells have the ability to self-renewal and differentiation. They require special culture conditions like feeder layers and growth factors. Stem cells are widely used in regenerative medicine, disease modeling, and drug testing. However, embryonic stem cells have ethical issues, which is why iPSCs are becoming more popular. Animal cloning means producing genetically identical organisms. There are two types: reproductive cloning to produce whole organism and therapeutic cloning for medicinal uses. The main method used is somatic cell nuclear transfer (SCNT). A famous example is Dolly, the sheep, the first cloned mammal. Cloning is useful in livestock improvement and disease research. However, it has limitations like low success rate, abnormalities, and ethical concerns. In SCNT, the nucleus of an egg cell is removed and replaced with the nucleus of a somatic cell. This reconstructed cell is then activated using electrical or chemical signals to start division. The embryo formed is later implanted into a surrogate mother. For successful cloning, proper synchronization of the donor cell cycle is very important. One major challenge is epigenetic reprogramming, where gene expression must be reset correctly. The efficiency is very low, usually less than 5%. Transgenic animals are those animals which have a foreign gene inserted into their genome. The gene is called a transgene. It is introduced using methods like microinjection, viral vectors, or embryonic stem cell techniques. These animals show stable inheritance of their inserted gene and can express it in specific tissues using promoters. This concept is very important in biotechnology and is frequently asked in exams. There are different methods to introduce genes into animals. In pronucleus microinjection, DNA is directly inserted into the nucleus of a fertile egg. Retroviral vectors are also used by the DNA insertion randomly. CRISPR or Cas9 is a modern technique that allows precise genome editing at specific locations. It is highly accurate, but still some limitations like unintended mutations. This topic is very important for modern biotech questions. Transgenic animals are used in biofarming to produce useful proteins like insulin and antibodies. They are also used as disease models to study conditions like cancer and neurological disorders. In agriculture, they help in improving growth and disease resistance. A well-known example is the onco mouse used in cancer research. They are also used in xenotransplantation, where organs from modified animals are used in humans. Stem cells and differentiated cells differ in many ways. Stem cells have high potency, meaning they can form many types of cells, while differentiated cells have limited function. Stem cells can divide continuously, whereas differentiated cells have limited division capacity. Stem cells are undifferentiated and have plasticity, while differentiated cells are specialized and less flexibility. The key idea is that stem cells maintain a balance between self-renewal and differentiation. Induced pluripotent stem cells. iPSCs are formed by reprogramming normal somatic cells into a pluripotent state. This is done using various factors. The advantage of iPSCs is that they do not have ethical issues like embryonic stem cells. They are useful in personalized medicine and disease modeling. However, there is a risk of tumor formation, which is an important limitation. Large-scale cell culture is done using bioreactors like stirred tank and perfusion system. These systems help in growing cells in bulk for industrial purpose. Different modes like batch, fed batch, and continuous culture are used. Important parameters include pH, dissolved oxygen, and agitation. During scale-up, challenges like shear stress and oxygen transfer limitation occur. Proper control of these factors is necessary for efficient production. Contamination is a major problem in animal cell culture. It can be caused by bacteria, fungi, mycoplasma, and viruses. These contaminants can enter from media, serum, environment, or even from the person handling the culture. Contamination affects cell growth and can give wrong experimental results. It can be detected using microscopy, PCR, and biochemical tests. To prevent contamination, strict aseptic techniques like filtration and use of antibiotics are necessary. Hat medium contains hypoxanthine, aminopterin, and thymidine. Aminopterin block the normal pathway of nucleotide synthesis, so cells are forced to use the salvage pathway for survival. Myeloma cells cannot survive because they lack HGPRT enzyme, and normal B cells die naturally after some time. Only hybridoma cells survive because they can have both immortality and a functional salvage pathway. This selection process is very important for monoclonal antibody production. The growth curve shows how cell number changes with time. The x-axis represents time and y-axis represents the logarithm of cell number. The curve has four phases: lag, log, stationary, and decline. In the log phase, cells grow rapidly and the growth rate is maximum. In stationary phase, growth stops due to nutrient limitation. One important concept is that the slope of the log phase represents the growth rate, which is often asked in exams. Doubling time is the time taken for a cell population to double in a number. It is calculated using formulas include logarithms. Shortcut formulas displayed in the slide. In exams, questions may ask to calculate doubling time or compare growth rates of different cells. A common mistake is using incorrect log values, so carefully calculate is important. Usually, only basic numericals are asked. This slide compares three types of stem cells. Embryonic stem cells come from embryos and are pluripotent. iPSCs are derived from somatic cells but also show pluripotency. Adult stem cells are found in tissues are multipotent. Ethically, PSCs are controversial while iPSCs and adult stem cells are not. Tumor risk is highest in ESCs and lowest in adult stem cell. In exams, Comparisons like this are very important and often asked. Microcarrier culture is used for adherent cells and requires beads for attachment. Suspension culture is used for non-adherent cells and does not need any surface. Both methods are used in large-scale production, but microcarriers help in growing adherent cells in bioreactors. One important point is that microcarriers act as a bridge between lab-scale and industrial-scale culture systems. Mycoplasma is a special type of contaminant that does not have a cell wall. Because of this, it is resistant to many antibiotics and difficult to detect. It can affect cell metabolism and gene expression. It cannot be seen under normal microscopy. So, special methods like PCR and DNA staining are used. Prevention requires strict aseptic conditions and proper filtration. This is a very important exam topic. Serum containing media has undefined composition and may vary from batch to batch. It also has a high risk of contamination. However, it supports good cell growth because of growth factors. Serum-free media has a defined composition and gives more consistent results. It is preferred in industrial applications despite being expensive. This comparison is commonly asked in exams. In SCNT, the nucleus is main factor that determines genetic identity. The process involves removing the nucleus from an egg cell and inserted a donor nucleus, followed by activation. One important point is that even though cytoplasm plays a role, the DNA comes only from the nucleus. Failure can occur due to improper gene expression. This concept is often tested in exams as a tricky question. Gene expression in transgenic animals depend on promoters and the site where gene is inserted. Tissue-specific promoters ensure that the gene is expressed only in certain tissues. Factors like copy number and position effect also affect expression. Reporter genes like GFP and LAC-Z are used to study gene expression. A key exam point is that the same gene can show different expression depending on its location in the genome. This slide gives a quick summary of the most important concepts from this unit. It highlights that SCNT is the main method for cloning. Hybridoma technology is used for monoclonal antibody production, and microcarriers are used to grow adherent cells in suspension. It also reminds the iPSCs are formed by reprogramming somatic cells. The slide also points out key comparisons like anchorage-dependent versus suspension culture and ESCs versus iPSC. It clearly mentioned that topics like growth curve, hat mechanism, and CRISPR are frequently asked in exams. So the slide is very useful for last minute revision. This slide contains very short one-line points for quick revision. For GAT-B, hat medium selects HGPRT positive hybridomas. iPSCs are reprogrammed somatic cells, and mycoplasma is an invisible contaminant. These one-liners are very useful for remembering key facts. It also highlights the most commonly asked topics like growth curve phases, hybridoma mechanism, and stem cell differences. The main advice is to focus on understanding mechanisms and comparisons. Are these most important for exams? Subscribe Brainzz and stay updated with the latest GAT-B 2026 preparation tips.